Alright, so this experiment is experiment 23. It's about the hydrolysis of polysaccharides. Alright, so the big thing that we have to kind of tackle today is this carbohydrates. So the definition of a carbohydrate is compounds composed of carbon, hydrogen, and oxygen in a 1 to 2 to 1 ratio. So as you can see here that we have a carbon. So you can kind of see it in the name. It's carbo. So you got a carbon, and then a hydrate means water. So you have water here. So for every carbon, there's one water molecule. That's what that is N means. So if you have N equals 6 here, as it is in glucose, if N equals 6, you have C6H12O6. And so that's a glucose, and this is what a glucose molecule looks like right here. And this is what a lot of sugar molecules look like, in that they have an oxygen in this top right corner right here, and then you go around this this chain right here, and you have carbons at each of the connection points. So if you have 6 carbons, then that's your carbohydrate right there. And they're usually in this ring structure like this. But they can go into a linear structure, and that's kind of more of an organic chemistry, biochemistry kind of approach. So the big thing here is that if you have a single sugar, so like glucose right here, it's called a monosaccharide, mono meaning one. So stuff like glucose and ribose, they're both monosaccharides because they only have one sugar. If you connect two, you make a disaccharide, di meaning two. So stuff like sucrose, lactose, maltose right here, those are all disaccharides. And the way that you connect them is through, you learned in 1201 this, through glycosidic linkages. You do a dehydration reaction, you take off one of these H2Os, and you connect them through this glycosidic linkage at that O right there. And so that's if you have a disaccharide. Now, if you have a few saccharides, like we have right here, where we have 3, 4, then you have a glycosidic linkage of 3 or 4 sugars, you have an oligosaccharide. And then multiple sugars, you have a polysaccharide, poly meaning multiple, and stuff like starch and glycogen and cellulose, those are all polysaccharides, because each one of these little circles right here is a sugar. So you have the link, amylase is another one. You have the linkage of a whole bunch of them, and these kind of web structures or wall-like structures or linear structures. If you have them linked together, it's a polysaccharide like that. All right, so that's kind of the general intro on carbohydrates. But now let's look at more closely at the polysaccharides kind of stuff. So this is the same picture that was on the last slide right here, and you have cellulose with that wall. Glycogen has this kind of big fiber like that. Starch a little bit smaller, but still a very netted web. And then amylase has this linear structure. So cellulose right here, the way that these sugars are linked like this, it's called a beta-1,4 glycosidic linkage between glucose monomers. So if you put a whole bunch of glucoses together, so this is a glucose, if we attach another glucose right here and then another one, and then another one and another one, and you can get this kind of web structure. You attach them through what's called a beta-1,4 glycosidic linkage, and that's just a special linkage that you'll learn about later. But you have to just know that beta-1,4 is part of cellulose. All these other ones are called alpha-1,4 glycosidic linkages. But cellulose is the major structural component of plants and other bacteria. We can't break that 1,4 glycosidic linkage, so we can't break down cellulose. So we can't digest stuff like corn or just like things that have a lot of cellulose in it. We can't digest that. Other bacteria can. So you talk about how cows can digest grass like that. They can break that beta-1,4 glycosidic linkage. We, however, humans and a lot of other life forms can break alpha-1,4 glycosidic linkage. So we can break down glycogen and starch and dextran and stuff like that. But yeah, so glycogen has this 1,4 glycosidic linkage, and again, it's through glucose. All of these right here use glucose as your monomers. Okay, and glycogen is the energy reserve of animals. We kind of have fat as our energy reserve. Adipose tissue, they have glycogen as their big. So we have fat and glycogen as our kind of big energy reserves in animals. Dextran is the reserves that microbes use. But again, it has alpha-1,4 and it's using glucose. And then starch is for plants. But again, alpha-1,4 and it's between glucose. So these are all your kind of polysaccharides that you kind of deal with in microbiology and other sciences. This is important with regard to the global carbon cycle. So what happens is that you have cellulases, or the enzymes that can break these glycosidic linkages. So I said that we have the ability to break down glycosidic, that alpha-1,4 glycosidic linkage is because of the cellulase enzyme. And these are extracellular enzymes, and humans produce them, thermophilic bacteria, aerobic and anaerobic microbes produce them. They're very ubiquitous, but they're good with regard to breaking down these carbons. And then once you break down the carbons into glucose, then you could metabolize it. We talked about the metabolism of glucose with regard to glycolysis, pyruvate oxidation, oxidative phosphorylation, and electron transport chain. We talked about that whole thing. So those then make carbon dioxide. Carbon dioxide then gets released into the atmosphere, and then plants can fix that carbon dioxide into sugar, and then put those sugars together into a polysaccharide like starch or cellulose, and then we can start this cycle all over again of then using these cellulases to break it down. And so these are very important enzymes with regard to the global carbon cycle that we have here. Amylases break down starch via glycosidic linkages. So amylases, so cellulases break down cellulose. Amylases break down these starch molecules right here. Humans have salivary amylase that you might have heard about, but bacteria have other amylases that again can be used to break down starch. Again, these are extracellular, and they break down glucose for the use of energy and respiration. All right, so what we did in this experiment was we tested for amylases. Is the first thing we did. So what we did was we used a growth plate that had starch in it. So we had an auger that had starch in it, and what we did was we plated microbes on it, and some of those microbes produces amylases. So they're going to break down that starch that's in that plate. So if they produce amylase, they will break down the starch around it, and they're going to create this like clearing that you can kind of see here. So that's the hydrolysis of the starch polymer. But we cannot see that naturally. So what we have to do is we have to add iodine to this plate. Once we add iodine, you can clearly see it. So you see how like over here the plate is like kind of like murky white right here and murky white right here. That's because the iodine didn't get over here, but there's starch all around here, and so the starch clearly went into here, but it didn't get over here. But you can see this zone of clearing because there's no starch right here for the iodine to go into. So yeah, again, the iodine binds to the starch and makes this kind of blue color that you see. And if there's an amylase, the iodine will bind to that starch and clear the zone of hydrolysis, and a clear zone of hydrolysis appears. So that's that zone of clearing like that. And then over here you see these other colonies right here. They do not produce amylases, and so no clearing occurred. So they did not use the starch that was in this plate as the source of carbon. And then we also tested cellulases. So what we did is we got what's called a Dubos plate, and we divided it into 4. We put a drop of bacteria in the center of the quadrant, and we put, so in the clear of the one, we put a drop of bacteria, and then we put different pieces of paper on each quadrant. So paper is majorly composed of cellulose. So if the paper was broken down, then you know that these microbes produce celluloses, and the paper each have different kind of concentrations or different types of cellulose, like concentrations or whatever it may be, just depending on where they got the paper from. So if the paper turns yellow, you can kind of see an older paper how it's like it got that yellow color. That's because bacteria have eventually broken down the celluloses in it. So if it turns yellow, then it's due to growth of the microbes. If the paper has more cellulose in it, then it will be more yellow. So you can kind of gauge how much cellulose is in the paper depending on how yellow it is. But then we also, so that's one experiment that some of y'all did, but then other ones did this one right here, that you got tubes of bacteria on them, and then you put the paper inside the bacteria. And so the microbes are going to be breaking it down in those tubes. So those are the 3 experiments that we did with regard to amylase, and the 2 different ones from cellulase. So the big takeaway here is that carbohydrates are very complex, and they can be broken down for energy. they can be polymers, monomers, oligosaccharides, or disaccharides. But we looked into polysaccharides in this experiment. The cellulose and the starch can be broken down with enzymes and then we make those glucose monosaccharides. And in the presence of these indications, this is kind of a general trend, right? That the presence of these enzymes can give an indication of the physiology of the microbes. So if they have the ability to break down these carbohydrates, then we know that they have a certain enzyme in their genetics to do that. And that's part of the physiology. And that's what we're kind of looking into the second half of this semester. So that's the experiment with regard to carbohydrates. And we'll move on to the next experiment after this. It's kind of awkward doing these like going out of the end of the video because I'm done talking, but I still have to say words because I ended on the wrong inflection. Anyway, sorry. That's it. That's it for this video.